Welcome to the BioInsights, where science comes alive.
Welcome to this explainer. Okay, today we are literally cracking a code. If you've ever stared at a massive biochemistry pathway and just felt like you were looking at complete alphabet soup, trust me, you are not alone. It kind of feels like an entirely alien language, right? But here's the secret. We're going to treat enzyme nomenclature not as some dry, rote memorization exercise, but like we're learning a highly logical foreign language. Because once you get the grammar down, everything clicks. And this incredibly complex subject suddenly becomes totally accessible. So here's our roadmap. We'll hit the molecular workhorses first, then dive into the two-name system, look at the 6 major classes, and finally untangle some really confusing enzyme name pairs. Basically, we'll start with the subjects, learn the grammar, master the vocabulary, and clear up those tricky false cognates. All right, section 1, the molecular workhorses. This is our introduction to enzymes. Before we learn the language itself, we really need to understand exactly who we're talking about here. Let's dive right in. Enzymes are the fundamental molecular workhorses of life. You'll hear them called protein catalysts and metabolic directors, but what does that actually mean for us? Well, out of the literally millions of energetically possible reactions happening inside your cells right this very second, enzymes are the things speeding up and selectively channeling specific reactions. And the crazy part? They do all this without ever being consumed themselves. By controlling what reactions proceed and exactly when they happen, enzymes direct the whole shebang, every metabolic event in your body. Think of them as the ultimate traffic controllers of biology. Without them, the chemical reactions keeping you alive right now would take thousands of years to happen. So yeah, they essentially make life possible by keeping everything running at the absolute perfect speed. Moving on to section 2, the two-name system and the rules of nomenclature. Let's see exactly how science assigns specific names so we don't just have absolute chaos in the lab. You know how a person might have a casual nickname that everyone uses, but then a highly specific formal legal name on their passport? Well, every enzyme gets 2 names too. There's a convenient recommended name for just everyday lab chat. That's the short one you're going to hear most of the time. But then there's the systematic name. This one is complete, entirely unambiguous, and used when precise identification is absolutely crucial in scientific literature. Having both lets scientists chat casually over coffee without losing the ability to be perfectly exact when they're publishing a paper. Let's look at that recommended name first. The absolute most common grammatical rule in this language is just attaching the suffix ase or ace to either the substrate being acted upon or the specific action being performed. Take glucosase, for example. It acts on glucose. Simple, right? Urease acts on urea. Or, if we name it by the action itself, lactate dehydrogenase removes hydrogen from lactate. Whenever you hear that ase ending, it's your immediate signal that you're looking at an enzyme that is actively doing work. It's basically the action verb of our biochemical sentence. But there are always exceptions, right? We call these trivial names. Enzymes like trypsin and pepsin were discovered so long ago that they just kept their original historical names. And, as you can probably guess, these give absolutely zero helpful hints about what they actually do, unlike our modern ACE system. They're kind of like the irregular verbs of the biochemistry language. You just have to bite the bullet and memorize them. But luckily, there really aren't too many of these legacy names left in everyday circulation. All right, section 3, the 6 major classes or the core vocabulary. Having covered the basic grammar, we are totally ready to learn the foundational vocabulary of the systematic name system. Here's the really cool part. All enzymes, every single one of those molecular workhorses we talked about, fall into one of these 6 major systematic classes based on the exact reaction they catalyze. It's like the ultimate biological filing cabinet. Everything from digesting your morning coffee to physically copying your DNA fits neatly into one of these 6 distinct categories. Let's build up your core vocabulary by walking through each of these classes. We'll take our time here because this really is the foundation for everything else. First up, oxidoreductases. These enzymes transfer electrons from one molecule to another. Just picture them as managing the cell's power grid, facilitating the oxidation and reduction reactions that shuffle energy around. Second, transferases. These guys act a lot like postal workers. They physically move entire functional groups, like a methyl or amino group, straight from a donor molecule to an acceptor. Third, hydrolases. These use water like a molecular scalpel. They break chemical bonds by forcing a water molecule into the structure, a process you probably know as hydrolysis. Fourth, we've got lyases. These are also bond breakers, but they do it without using any water.
They add or remove groups to form or break double bonds. It's essentially taking a structural shortcut. Fifth are the isomerases. Imagine rearranging the furniture inside a single room. They shuffle atoms around within a single molecule to create an isomer. The overall chemical formula stays the exact same, but the internal structure changes. And finally, number 6, ligases. These are the builders. They join two molecules together. It's a heavy lifting process, so it typically requires a nice energy boost from ATP. And that's it. Those 6 categories encompass the entire core vocabulary of human metabolism.
Section 4, Confusing Enzyme Name Pairs, the false friends. Now that you actually know the rules, we have to carefully shift our focus to the tricky nuances and false cognates where so many students get completely lost. Okay, let's look at our first pair of false friends. Listen really closely to the syllables here. Synthase requires energy specifically from ATP to drive a synthesis reaction. Because it joins things using ATP, it's technically a type of ligase. On the flip side, synthase accomplishes synthesis without any direct ATP consumption. It usually acts as a lyase or an isomerase. So synthetase uses ATP, synthase does not. It is a tiny linguistic difference, but man, it has huge metabolic implications, especially when you're trying to track the energy budget of a cell.
Here is another classic mix-up: phosphatase versus phosphorylase. The crucial thing distinguishing this pair is their underlying mechanism. A phosphatase uses water, just simple hydrolysis, to physically remove a phosphoryl group from a substrate. It's basically a subtraction process. But a phosphorylase uses inorganic phosphate as a tool to break a chemical bond. And in doing so, it generates a newly phosphorylated product. So to recap, one strips a phosphate off using water, while the other actively uses phosphate like a crowbar to break things apart.
Let's move on and see how this all builds into our final confusing trio for this explainer: dihydrogenase, oxidase, and oxygenase. These are 3 specific types of enzymes that actually all belong to the oxidoreductase class, meaning they all involve oxidation. But they behave fundamentally differently based on what accepts their electrons or precisely how they handle oxygen. Understanding this trio right now will save you endless confusion down the line.
First is dehydrogenase. In this reaction, hydrogen atoms are transferred, and molecules like NAD+ or FAD step up to serve as the electron acceptor. The absolutely critical takeaway here: oxygen itself is not involved in accepting electrons at all. This enzyme strictly moves hydrogens around to facilitate redox reactions, completely independent of oxygen.
Next up is oxidase. Now, oxygen actually enters the picture. In an oxidase reaction, O2 acts purely as an electron acceptor. It steps in, catches the electrons, and forms water or hydrogen peroxide. But listen closely to this distinction. The oxygen atoms themselves are never actually incorporated into the substrate molecule. They just act as a molecular catcher's mitt for the electrons, and then they leave the scene. Finally, oxygenase. This one is uniquely different because it doesn't just catch electrons. No, one or both of the oxygen atoms from O2 are directly incorporated into the actual molecular structure of the substrate itself. It permanently alters the molecular architecture by forcing oxygen right into it. It is a structural integration, not just a quick electron handoff.
So mastering these specific name distinctions is absolutely the master key to understanding biochemistry. Think about it. You've learned the subjects, the grammar, the vocabulary, and even those tricky false friends. This leaves you perfectly positioned to explore the intricate, beautiful pathways these enzymes actually control. You hold the linguistic key to human metabolism. So the next time you look at a massive, incredibly complex metabolic pathway, ask yourself, are you just seeing a random list of chemicals, or are you finally reading the hidden code of life?